Hey, welcome back. Last time we talked about proteins. We talked about proteins a lot, and so the key technique I'm going to introduce to you in this part of the semester is the technique of SDS page. And here it says membrane proteins and SDS page, but this is a technique which can be used for proteins isolated from anywhere in cells. Doesn't have to be membrane proteins, but it goes reasonably well here. So let's talk about that. SDS page stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. So let's break the long name down into pieces and look at the pieces. So let's start off with the first piece there. The SDS piece, sodium dodecyl sulfate, refers to the main chemical that is used to solubilize and treat the proteins that you want to analyze. So this technique is a way to separate proteins based on their molecular mass. And this can be a precursor to mass spectrometry, which we talked about before, as a way of partially purifying proteins in some senses. So we'll talk about the general technique in a moment, but I want to show you this molecule that's used to treat proteins prior to the electrophoresis part of this technique. So here is the molecule SDS, sodium dodecyl sulfate, and you can see it's a long hydrocarbon chain, just like a fatty acid, conjugated to a sulfate group here, which is often associated with a sodium ion in the chemical form. So we have a hydrophobic tail and a hydrophilic head. So that is a really good molecule for disrupting cell membranes and for denaturing proteins, because it disrupts a lot of hydrophobic interactions. So proteins stuck together by hydrophobic interactions will be broken apart into their separate subunits using this drug, and then the proteins themselves are heated up in combination with this SDS and a compound called beta-mercaptoethanol. And that beta-mercaptoethanol, if you walk into a lab, you may smell it. It smells like stinky eggs. Again, that's sulfur-based. So the SDS unfolds all the proteins, working with the heat and with the beta-mercaptoethanol to unfold the proteins. And then the SDS molecules here, which are shown as these fatty acid tails, these fatty tails with this hydrophilic head, they basically cover the protein when it unfolds. So it goes from this globular folded structure to being completely unfolded because of the SDS and the boiling and the heating and the beta-mercaptoethanol. Totally unfolds, and then the protein is covered in the SDS. And the SDS has this negative charge because of this sulfate group up here, and so that negative charge ends up being proportional to the molecular mass. So the bigger a protein, the more negatively charged the protein will become once it's covered in the SDS molecules. So here's what we do. Here's that's the molecule. Here's the kind of big picture of this technique. We take a sample, and that could be membrane fragments, it could be Golgi, it could be whole cell extracts, nuclear extracts, anything we like. We take our sample, and here we're looking at membrane fragments because we'd be interested in looking at membrane proteins. Here are membrane fragments that we've isolated from cells. We treat them with the SDS, and we treat them with beta-mercaptoethanol, and we heat them up. Then we take the sample and we put it into the well of this gel, which is shown in yellow. And the gel is kind of like the meat in a sandwich. So there are two glass plates, and between the glass plates is a gel made from polyacrylamide, so polymerized acrylamide. So acrylamide is a neurotoxin. It's not such a nice chemical. When people used to work with this and didn't realize it was a neurotoxin, their fingertips went numb over years after working with this compound, so you have to be careful with it in its powdered form. Once it's polymerized, it's perfectly safe. And so the polyacrylamide gel is sandwiched between these plates, and you have these little cutout wells in the top where you can load anywhere from 15 microliters up to maybe 30 or 40 microliters. So that's your sample that's been boiled up in the SDS and beta-mercaptoethanol. So what we then do is we attach the gel to an electrical source, to a power source, and we put the cathode at the top, that is the negatively charged terminal, and the anode at the bottom, and that's the positively charged terminal. Now, because the proteins are covered in SDS, when we turn on the power supply, the proteins will migrate through the gel towards the positive terminal, and so they'll separate out. Now, if you just leave this running for hours and hours and hours, it'll all just run off the bottom. But if you stop it at a certain point, all the proteins at the same molecular mass have arrived at the same point on the gel. So the distance from the gel well is inversely proportional to their size. So this protein at the bottom is the smallest one, and this protein at the top is the biggest one. And so we've separated out all of the proteins, and so on our gel here we would have all the proteins in our 5 samples separated out on the gel, and then we can use special stains and dyes to visualize the proteins. The most famous one is a dye called Kumasi Brilliant Blue, but you can stain with silver as well, which is very, very sensitive. And so when you stain the gel, you can see all the proteins. So here's a membrane fraction from human red blood cells, and these different bands are historically given numbers, and we know what the bands are now. These are components of the cytoskeleton. This is an anion exchange protein, which is important in the exchange of carbon dioxide and primarily oxygen from the red blood cell. And then there are some other proteins down here, another cytoskeleton protein and a metabolic enzyme, GAPDH. So all those proteins are separated by electrophoresis. Just to give you an idea of what this looks like actually once you've stained a gel, here would be all of the wells that run into the gel, and so these are called the lanes, and each of these different strips is a protein of a different molecular mass. And over on the right here is what we have, our molecular mass markers. So this one here is telling us that at this position on the gel, if we draw a line across this position on the gel here, all the proteins here have a molecular mass of 35 Daltons, 35 atomic mass units, 35,000 Daltons, sorry. So that would be the same as 35,000 grams per mole. And so 25 is down here, 15 is down here, so small at the bottom, big at the top. So that's SDS page. It's a technique for separating proteins, and then we can use these separated proteins to do numerous other techniques, and those techniques will come up a little bit later in the semester. So that's it, just a quick technique for looking at a way to separate proteins in complex samples and visualize them. I'll be back next time when we're moving into a new chapter.